you some introduction or basics here, that we are going to use the next generation sequencing data that is being produced from the high throughput sequencing platform. And as I use the term high throughput sequencing platform, they can also provide or incorporate the errors in the sequences which we are getting out from the platform. So there is need to first analyze the raw data which we receive from the sequencing platforms. I will give you some brief introduction here how a sequencing platform works. In the case of Illumina, here we can see that a genomic DNA is extracted and then it is shared into certain fragments of the DNA, and then the adapters are attached to the ends of these sequences, and then there is the amplification of these sequences or template sequences inside the chip, and then the next step is the sequencing by synthesis. So each nucleotide has the fluorophore labels there, and then when the laser comes there, when they attach there, and then the laser comes here, they are being detected. And during the detection, how the quality is measured. So this is during this step, at the base call step, the quality is being measured. Suppose how much is the signal intensity there, which is being detected there, and that is the basis of the quality of that particular base incorporation or during the base call. I will give you more details about the quality of the sequences which you receive there. Okay, so this is the process where we get the sequences out of this high throughput sequencing platform. So the technologies are different, like the Illumina, there is ABI. So they all use different types of technology, like different types of sequencing mechanisms. But at the end, what we get here, the quality values for each of the nucleotide bases. So how we can analyze this? When we get the data from the sequencing platform, we receive the FASTQ files most of the time. Sometimes we get the raw data out of that, but most of the times we get the FASTQ data. Okay, so how the FASTQ data looks like, you can see here there are 4 lines for a single sequence. This is the header of the nucleotide sequence, then the nucleotide sequence here, AAGGCT, that is the nucleotide sequence, and then you can see the third line here which starts with the plus sign. Okay, that is the quality header. Most of the times you will see like only plus written here but nothing else, but it should be the same as the sequence header here. Okay, then the fourth line of that sequence, this sequence, it is the corresponding values, quality values for each of the nucleotide positions for this particular sequence read. Okay, so this is the FASTQ format data. You might have gone through the FASTA format, so where you see the greater than sign there, and then you see the FASTA header, and then followed by the sequence. In the FASTQ file, there are 4 lines. First is the FASTQ header here, sequence header, then sequence, quality value, quality header, and then the quality values here. So what are the quality values here written, like A, B, B, each of the corresponding nucleotides there in the sequence. So how we interpret this quality value? So there is the quality value interpretation. We can see here the Fred quality score. They are measured in the Fred quality score. I think I'm audible to you. Yeah. So yeah.
Yes, sir.
Yes, sir.
Okay. So the quality values are measured in the Fred quality score units. It is the quality identification of the nucleotide bases or nucleobases generated by the high throughput sequencing platforms or the automated DNA sequencing platforms. This is like the Fred quality scores are assigned to each nucleotide base call. I showed you there. They were assigned to this step here when the base call was being performed. So the quality values were given there, like, okay, signal is good, high quality value. If it is not good, mixed signal, quality value is low. If there is no signal, it means it was nothing there. So like that, something wrong had happened, like mixed signals were there, so not good quality. So during the base call, Fred quality scores are assigned. So how we see the Fred quality scores when we interpret it they are the logarithmically linked to the error probabilities, right? If the quality score is, Fred quality score is 10, it means that there is the probability of incorrect base call 1 in 10. So one base is incorrectly incorporated there. Like 10% is the error if the quality score is 10. The same way 20 base call, quality score means 99% accuracy. 30 Fred quality score means 99.9% accuracy, and likewise up to 60. So what we get in Illumina, that is the 40. Why? Because we get, or Illumina also officially they say that we have 0.01% of the error rate in during the sequencing. So we get the maximum Fred quality score from the Illumina, that is the 40 Fred quality. Is it clear? So how this ABB I showed you in the FASTQ file here, this one, here the plus value is for the quality header, and then the quality basis, quality values here for each basis. So ABB, how we convert it to the Fred quality 10, 20, 30 or 40. So that is done by here. If you see the different encodings, Fred quality score encodings being used over the time. So you see here the Sanger quality score is Fred plus, yes. Do you want to say anything? No. Okay. So you can see here the Sanger Fred plus 33. So what does it mean? Plus 33. So let's say plus 64, Fred plus 64. So this is the ASCII scores of these characters. Like exclamation has the ASCII score of 33, right? So the values which are being interpreted here, they start with the 33 here. So that is Fred 33. If the scoring is starting from Fred 64 here, like this, 0, 1, 2, 3, 9. So this is the Fred scores, but don't get confused with the ASCII score, like the A, B, C, D. So these are the encodings, and then they denote for each of the different encodings for different quality values. Okay, so in the Illumina, you can see Illumina 1.8 plus, that is this one. They starts from 0 to 41. Sometimes we also get the Sanger, this one 0 to 40, Illumina 1.3. So this is the 0 to 40 score, and it starts from the Fred plus 64 here. Okay, so the encoding is plus 64. The characters which will be encoding the Fred values, they will start from ASCII score 64. Okay, yes.
Sir, would you please pardon FAT K, the quality value? Actually, I haven't understood this one.
The quality value, Fred quality value.
No. FAT, what is the full form of FATQ quality value? What is the meaning? What it means?
Fred quality score.
No, sir. FATQ quality value. Just now,
FASTQ. This one here. So these are the quality values here. Okay, and they are written here in certain encodings. Okay, like this character A, B, B. So they are written here in a encoding. If you see this one here, come back here. So the value here is the 65. Okay.
Sorry, excuse me, sir. Sir, FATQ I am asking. FATQ quality value. Just now, whatever you are telling. FATQ.
FATQ.
What is the
actually? It is written. You have written
okay,
FATQ.
Okay. Sorry.
This one, yes.
It's
Yes.
typing error. I'm really sorry about that. FASTQ.
That's why I thought it. What is this? This is the new form. I thought it.
No. It was the typing error.
Okay, thank you, sir.
So what are the first 4 lines, like the letters in the first 5 lines?
These are.
Yeah, sir.
Okay, so this is nothing. They are just the Illumina. This is telling us that it is denoting for the Sanger encoding. This is for the Solexa, which starts from 64. Okay, plus 64. Fred 64, Solexa plus 64 starts from here. Okay, and these are the ASCII characters. If you see here, these are the ASCII. These are the characters, and they are converted to the ASCII values. That's it. And that's how the Fred plus 33, Solexa plus 64, the encoding names were given based on these,
So
the value of the ASCII
we
codes.
Excuse me, sir. All these are the raw reads, like these 4-5 lines above.
These are not the raw reads. These are the encodings here.
Okay.
Okay. For the
so
Illumina, yes.
typically a FASTQ file will have all these encodings, like for all the systems.
Okay.
So let's
So
say
depending on if you receive any FASTQ file, so there is the possibility of you get the Fred 64 encoding sometimes. Sometimes you get the Fred plus 33. Okay. And sometimes what happens, you also get the Solexa 64, like the older data. If you get any old data set, so you might get the 64 encoding there also. Okay.
okay.
So the encoding is different, but the quality values which they represent, either they start from here at the 33 from exclamation till the I. Okay, they will be starting from 0 to 40. They start from anywhere on this scale here, from any character. They will last till the 40 for Illumina dataset. Okay, but for the, I think it used to be for different Fred quality scores for the different platforms, right?
So
So
for Solexa, the Fred quality score can also be -5, is it? Why
for
does it say?
yes, so Alexa is -5. Yes.
Okay.
Okay, so this is, we have seen how the encodings are used, different types of encodings are used to assign the Fred quality values or Fred quality score for the FASTQ quality basis. So this is just a table where you see the ASCII code, which I showed you earlier here, the exclamation sign, okay, character exclamation, and it has the ASCII code of 33. So if you say any Fred plus 33, you might find the exclamation sign here, sorry, exclamation sign, which starts from 33, and it will go up to I, up to 40. Okay. So that is how the encoding is done. This is just for the information. We also use the paired end sequencing data. So the sequencing data can be produced either the single end or the paired end. In the single end, we sequence it from one direction. But in the paired end sequencing, first we sequence it from the one
Excuse me,
direction
sir.
and then in the reverse direction. Yes.
Sir, that lines are representing the raw reads only with the next with the types of next generation sequencing that types only given, no?
This one.
These lines are raw
No, no,
reads
no,
with
no, 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 no. These lines are just showing the different types of Fred encodings used. Okay.
different scores that are
Yes,
indicating.
encoding. Encoding means what codes are being used to assign the Fred quality values. Okay.
To the raw reads,
Yes,
scores assigned
raw
to the
reads or any FASTQ dataset should have some kind of encoding or codes, that those
and,
codes will assign the Fred quality values. Okay.
and these are types of NGS also assigned to that Fred score, data generated from that type.
Yes.
Okay, fine, sir. Thank you so much, sir.
Okay, so we have either the single end sequences or the paired end sequences with us. So in the case of single end sequences, that is sequenced from one direction only. That will be, or we call it like forward sequences only. When we do the paired end sequencing, we receive the reverse sequences also, or reverse reads also. So when we get the reverse read or paired end sequences from forward and reverse, then there is the benefit over the single end sequencing, that we can assemble the genomes better, and then we can also perform the repeat region or resolution of the repeat regions better during the genome assembly process. Right. So there is the certain benefits of using the paired end sequencing outputs. So there are various tools available for the quality analysis of the raw data as well as the process data. Once you process the data with any tool, you would be interested in checking the quality again, whether the quality has improved or it is the same. Okay, so you can use those tools like we will be using today here, fast QC. One of them is there. So it tests or checks the quality of the data and shows us the in the format of different graphs. Okay, so to install the fast QC, you can go to this website also and then check more details about this there. I will show you in short. You can install the fast QC in your system in the Conda environment using the Conda install -c bioconda and then the fast QC. Or you can also use the sudo apt install fast QC. So you can install either any of these two methods for the fast QC. Fast
Conda will
QC,
work on any software.
yes, Conda is the software. It is you can install the Anaconda inside the Linux. Okay.
Will work on any platform.
For the Linux, you can use Conda. You can also use it for the Windows, but you won't be able to use it the same way as we use in Linux. Okay, so you can also use the fast QC in Windows operating system. There is the you can say exe available. You can download and install it in the Windows operating system. Only the fast QC. Okay, so this is the command. I will show you everything in stepwise manner. Then we will use the Trimomatic for the trimming and filtering of the data based on the output from the fast QC. The graphs we will be seeing there, the quality of the data, and then we will process the data using the Trimomatic. And there are other tools also available for the quality filtering and analysis. So one of them is the Prinsic, other one is the adapter removal. So Prinsic has coupled both the outputs. You can check the data and then trim the data for the good quality. Adapter
Hello.
removal works the same way. Yes.
Sir, all these are packages within Conda itself, Printsec, Sixcorer.
It is not available in the Conda. You can install it in the
Yeah,
Conda
means
environment.
we can install it. There will be packages are there.
Yes, you can
Okay.
install it. Yeah.
Thank you.
Okay, so there are different tools available, like cut adapt is used to remove any of the adapter sequences which are present during the after the base calling, and then you get the fast queue files. You will get this. If there are adapters, you can also trim those adapters there. Okay, so this was the some of the basics I wanted to show you, and I will share the screen of the.
Please share this PowerPoint also with
I
us.
will share it here. Yeah, I will share it with you.
Conda requires Python installed.
Actually, Conda is Python based, you
Okay.
can say, and it tackles most of the times when you have like multiple dependencies or different types of dependencies for different softwares or tools. So what you can do, you can create an environment there and use one tool in that environment, and that will not clash or make any trouble with other dependencies. Like you have numpy version something and the other tool requires numpy of version some other versions. So that is the best thing you can do here. Inside the Anaconda, you create the environments and install the tools for each of them separately, and that they will never get clashed. Okay, you can also do the virtual environment thing in Python the same way to avoid those dependency errors. Okay, so let me share the screen. I think it is shared. Okay, let's first show you the other website there. So here, so this is the fast QC website. The link is there. You can download it and install it. Like you can go to the download, and then you can get the different versions, or you can just type the command which I showed you there. Okay, you will also get here the, sorry, there is the fast QC, this one, fast QC. You can get the Windows, Linux zip file, Mac DMG file. So different versions you can get here for different operating systems. There is another tool, Trimomatic, which we will be seeing today. It is available here from this link or from this website. You can get the manual here for the print sheet. And go all the details about the print sheet if you want much details or deeper details of the Trimomatic tool. Okay, so the next thing, there are certain adapters being used during the sequencing. And suppose you have got the data from NCBI SRA database and you don't know what are those adapters were used. So you can do one thing here, come to the Trimomatic GitHub page. The link is there. I added that. So you can come to this page here and then you can go to the adapters directory and download all these sequences here. So these are the Nextera, TrueSeq. So these are the adapter sequences which were added here, and you can download them and put them in a single file. Okay, I will show you, and I rename that as all adapters.far. Okay, so for your clarity, I'm showing you how you can create this file here for the adapters.
So won't the adapters that we use will also be differing according to the sequencer that we used?
Yes, definitely. So the thing is here that when we use the adapter sequences, they are the Illumina adapters. Okay, and different kits have different types of adapter sequences, like TrueSeq, Nextera. So they are different kits available for the sequencing. So they use different sequences for the adapters. Okay.
So what we're seeing here is suitable only for Illumina, like not PassBio or anything else. Right?
Not really. Yes.
Okay,
For that
thank
reason, you need to get the adapter sequences for that particular sequencing run or that particular sequencing
you.
platform.
Okay, thank you.
Excuse me, sir.
Yes.
Can you please show that website where you showed all the different types of adapters?
Okay. This is here. Let me share the link.
In the GitHub.
Yes, on the GitHub.
What link is this, sir?
This is here. I shared it. You can just open it now.
This is a link for the Trimomatic.
Trimomatic, you can get the source here, or you can also get here. Yeah, latest release. Okay.
So these adapters will be universal based on the then if we know what the company is sequencing in, we can take the respective adapters from this website.
Yes, you can do that. If you don't know, you can just use all of them. And if during the Trimomatic, when you are going to use that, you will find it there. If the adapters, these adapters are present there in the sequences, they will be trimmed out. Okay.
Okay.
So let's go back to the fast QC then. So let me share the screen. Okay. So the data here, I will show you. Here is the forward read and the reverse read. I told you about the paired end sequences. So this data is in the paired end format. I downloaded it from the SRA database from the NCBI and renamed it as the name of the bacteria and then the forward, and subsampled it to the 100,000 reads to do the process fastly. So this is the forward read and the reverse. Sometimes you will see 1 and 2. So 1 means the forward read and the 2 means the reverse read. If we open this file forward, this is the FASTQ file. You will see the same format. Header, sequence. Then the third line is the plus, that is the quality header, and the fourth line is the quality value, that is CDDFF. So this is the encoding which was used during the base call. right? Oh, sorry. And you can see here for the forward file, you see one written here, which means this is the forward sequence file, forward sequence. If you see the reverse file, you will see the two written here, means it is the reverse sequence file. Okay. So we will run the fast QC first. For that, we will create a directory, output directory. So mkdir fast QC out. We can rename it as fast QC raw also. Anyway, this is the fast QC out we have created. So output will be saved inside this directory. Then we will run the fast QC minus help. So there are various help or the options available in the help. You will see there is a lot of options available for the fast QC. You can run fast QC using different options. Suppose you have the cassava output data. That is a tool. So if you have this cassava output, you can give this flag and it will use the data as it is coming from the cassava. It will recognize it. If we have the nanopore sequences with us in FAST5 format, not in the FASTQ format, we can give this flag here or option to tell FastQC that this is a nanopore output we are giving you. So we can use the nano option here. No filter if running with cassava, then don't remove read flags. So if the cassava is there, you can use this filter also. Extract if set, then zip output file will be uncompressed in the same directory. So this is about the output. Java. So these are the options you can read through, and if you read to use it, you can apply here. Like the minimum length, set as artificial lower limit of on the length of the sequence to be shown in the report. If you don't want to consider sequences less than certain length, you can give the minimum length option, because at the next step you will remove those sequences which are shorter than that particular length. The format, if you want to provide the format like BAM, SAM, BAM map, SAM map, or FASTQ, so you can use -F to provide the particular format file you are giving as the input. Number of threads you can use to parallelize the process, and that will speed up. Suppose you have 8 threads available or 16 threads of the CPU. Okay, so you can provide this thread option to increase the processivity. If you have contaminant file, you can also provide here using the -C. So these are the options you can all use. List of adapters, sequences you can also provide here. So FastQC will check the specific adapters there if they are present. Otherwise, it will use the default adapters which were added to the FastQC. Right? So this k-mer based operation you can also apply here using the -K and then provide the length of the k-mer bases there. So these are the few options I have showed you of the FastQC. To run the FastQC, we can do here FastQC. We will call the FastQC tool and then -out, that is for the out directory, -O or -out dir. FastQC out, the directory which we have created just now. So that will be the output directory name. Then we will give the input sequences, forward and reverse. If you want to use the threads, you can also give here -T or -threads 4. As 4 is available, I have given 4, and this should be -O or -out dir. Right? We will run this, and it will process the data, analyze the sequences present here, and then give us the output in the output directory. And what those outputs are, we will open that directory and check the result. It is done. ls fast QC. So we have here 4 outputs for these 2 data which we have generated. So there is one zip file and the other one is the HTML file for the forward and reverse. We can open this HTML file in any of the web browser like Chrome, Firefox, or Microsoft Edge. Any of the browsers can be used to open these HTML files. So let me open that, and this is present in this particular directory here. Okay, so you can navigate to this place and then open the HTML file. As I am using the WSL here, so I will open it from the Windows side, going inside the fast QC directory and then open the forward and reverse file for the HTML. I'll share the screen. Okay. So you can see here, close this. This is the forward file you can see, which was generated from the fast QC tool. Okay, these are the different sets of graphs and statistics which we have received here. The same graphs and stats were given to given as in the output from the for the forward and reverse file. So I will explain it to you. This is the basic stats. You can see file name, the data which we used as input. So this is the forward 100,000 fast QC. File type conventional base call, and encoding is Sanger Illumina 1.9. Total sequences were 100,000. Sequence length 35 to 250 base pairs long. Sequences available. Percent GC of the data is 41. Okay. And we also see the per base sequence quality here. The second plot here. What it's telling us, on the y-axis, this is the Fred quality score, starting from 0 to 40 for the Illumina 1.9. Right? So this is the Fred encoding 1.9 Illumina. Okay. And y-axis is showing the base position for all of the reads throughout the inside the forward read data. All of the read positions here, starting from 1 to 250, right? And for each base position from 1, 2, 3 till 1 250, what is the quality value here, which is showed by the box and whisker plot here. So these boxes are showing the 25th to 75th percentile of the quality values for the data set, for the forward data. These error bars are showing from the 10th to 90th percentile of the data set. And red line here, you see the red on the top of the bar boxes, they are the median values for the Fred qualities. And the blue line you are seeing here, the mean quality values for the each base position, right? So what is the mean base position? Like if there is 100,000 reads, for all of the 100,000 nucleotides bases present at base number position 1, the mean quality is around here, 33 point something. Okay, this is the mean quality. Median is around 34, right? Is it clear, this plot? So the quality seems perfect here, not very bad. The last here you can see at the trailing end, or the at 3' end, you can see the drop in the quality, but it is still good. And we can consider using the Fred quality 20 if you are working with the Illumina dataset.
Excuse me, sir.
Yes.
What is there in the x-axis? What it denotes?
This is the Fred quality value, 0 to 40. sorry,
Sir,
y-axis.
x-axis.
X-axis is the base position.
Base position of the sequences.
Sequences. So suppose there are 100,000 sequences available. Okay.
Okay.
So throughout the 100,000 sequences, base position number 1. So that will be calculated for each of the bases.
Okay. Okay. So
Throughout
it is
the
kind of
100,000.
summary of the all the
Yes.
sequences, right?
Yes. Yes. Right.
Okay. Thank you.
Okay. So the next one is the per sequence quality score. You can see on the x-axis, this is the mean sequence quality. That is the Fred score starting from 2, or you can say 1, till the 38 here. And the quality is increasing. It's going up, like very few sequences have less than, here you can see 28. And then when you add the number of the sequences in the bucket, the quality increases and goes
Excuse
up
me,
to
sir.
38. Yes.
Sir, what is the mean value and median value which you told in the red line and blue line? What does it mean?
So the mean value is here, okay, out of 100,000 sequences, okay, what is the mean value of the mean Fred quality score of that particular base position? That is the mean value, right? That suppose there are 10 people, okay, and their height is something. So what is the average height of 10 people? The same way, if there are 100,000 bases present there, what is the average quality at that particular position? That is the mean. The same way you can calculate the median value here, right? So it tells us if it is better, higher than 20 mean, we
Yes,
can
so
accept the
it's
data.
an indication of that score only, nothing else more.
This is the score, yes, Fred quality value. Yeah. Okay, so the same way, the same data, but it is shown differently. Like if you add the number of the sequences, how the quality is present there. And you can see the majority of the sequences have quality better than, you can see more than 5,000 sequences. Less than 5,000 sequences have quality less than 35. Rest sequences has the quality which is higher than 36. Okay. The same way per base sequence content here. So it shows us the error. Here the green tick shows that it is good, it seems fine. But the red cross here shows that there is certain problem with this analysis or this output. So what is the problem here? You can see the per base sequence content doesn't seem good at the terminals from the 5 prime here this side or from the 3 prime side. So what needs to do, because some of the bases are over represented and under represented here, percent %ATCG. So in that case, what we should do, we should trim this particular region from this side, from the 5 prime, and from the 3 prime, this particular region we can trim. So we can trim up to 19 base pairs here from the 5 prime, and 240 will be the total crop of the sequences. So to remove after this reason, right? So this we have selected here that this needs to be sorted out. Then,
This is the fast QC after Trimomatic.
pardon, I can't hear you clearly.
Yes, sir, this is the fast QC after Trimomatic.
No. This is for the raw data. We will run the Trimomatic after.
But it shows adapter content tick. That's why I asked.
It shows what?
Adapter content tick like that.
Yeah. I will come
That's
to
why
that.
I asked. Okay.
I'll come to that. Okay, so per sequence GC content is here. You can see it shows the warning sign. Like per sequence GC content has some issue. But what is that issue? This is the hump here which you are seeing, like a second peak. But it doesn't seem wrong to me, because you see the highest peak is showing the same range as the theoretical distribution of the data set if it is there. So we can accept this one as the GC content. The GC content is important in the case of, suppose you have a bigger peak here, like majority of the sequences have a second GC content or GC percent peak there. So that might indicate the chances of the contaminations during the sequencing or during the library preparation or during the isolation of the organism. Like any of the steps, they might get contaminated. So that might represent the contamination in the data. If you see the majority of the sequences, they are showing a second peak there, right?
Hello.
Yes.
Sir, I have a query.
Yes.
The GC content per sequence shows the GC percentage of base pair, right? G and C. So
Yeah.
in the same way, is there anything which shows 80 content?
You can calculate it, right?
Okay.
Okay. So per base N content is there. Actually, this is being used most of the times, like this is used in the taxonomic analysis also. It is being used by the taxonomists because GC content is highly conserved for different species. Okay. And that's why the GC content is being used as finding out the contaminations if they are present there. We can even separate the different species of the same genus, right?
Sir, I have one question. If a gene of interest is of high GC content, so and Fred score is related to GC content, all right? So
No,
when we are
no. Fred, if the GC content is high, then Illumina has sometimes the higher error rates, but it is
so,
not related.
sir, but the thing is, when we'll be performing these quality parameters and everything, our gene of interest or the sequence will be it will be separated from the rest of the data set, all right? But my gene of interest will be in that low quality score. So what will be the optimal strategy we can go about
For the,
for extracting those GC rich genes?
I can't understand your question. Suppose you have the whole genome with you.
Yeah, and we are performing de novo assembly. Let's
Yeah.
say, for example, for a particular bacteria,
Mhm.
so it has one particular operon which is high GC content. It has high GC content, all right?
Mhm.
So when you will be doing normal Illumina, so during this Fred score and all the quality parameters, since it is high GC content, it will be removed. It will be in separate data sets. So whenever
So you will be getting the CDS there, okay? Complementarity determining sequences, okay? CDS or ORS we can say. So what you can do, you can check the content, GC content of each of the ORFs there, and which region in the genome they are coming from. You can get it from the GFF file, and you can say, okay, this particular gene has separate GC content from the rest of the genome or rest of the genes.
sir,
Okay.
but how will I be align that, assemble that in a particular genome?
Okay, that is the separate issue, and okay, which is not being included here. Okay.
Okay.
Yeah. So okay, I was at the GC content, so it seems fine here. Per base N content, you can see there is no N's available, but most of the times when we get the Illumina dataset, we see the N's present there. Okay. So if the N's are there, they will be removed out during the Trimomatic run. Sequence length distribution. So this plot is showing here sequence length starting from 35, 32, 251, and most of the or the majority of the sequences, they are in the range of longer than 234 to 251, 250 base pairs, and very few sequences, I think they are very nominal present there as less than 230 or 235. Sequence duplication level. So this plot is showing here, this one. If the sequences are exact duplicates there, and if there is high number of the sequence duplication, it means that the library wasn't well prepared, or the sequences are not very good. So in that case, you can remove those exact duplicate sequences before the assembly, but it is based on the downstream analysis what you are going to do. If you are going to do the assembly, you can remove those duplicates there. Otherwise you can keep them, they are not going to harm during the assembly process. Only then thing, you can reduce the data and then save the computational resources. But if the data, if you see more or high number of the sequence duplicated in the sequence duplication level, like more than 10, 50 or 100 or 10,000, then you can remove those sequences there. Okay. Percent of sequences remaining in deduplicated. So like 96,000 sequences will be remain there if you remove the duplicate sequences. Overrepresented sequences are presented. So this might be an overrepresented sequence, this particular present in 175 reads, and then the percentage is 0.17 of the 100,000 sequences. And the possible source, it may be the TruSeq adapter index 1, right? So what we can do now, like we are going to use the adapter sequences to remove the adapters which are present there. Adapter content, if you see any of the adapters present there, they will be shown here. How much percent is the adapter present there? As we see, this is the TruSeq adapter here for the from the Illumina, but it is like 0.17%. So which will not be shown here properly, and it doesn't have many adapter sequences. So I think this plot is like this output is clear to you, right? Do you have questions?
Yes, sir.
Yeah, please ask. We need to get a little bit faster. Yeah, so you can ask.
Hello,
Yes.
sir. Sir, can you go to the overrepresented sequence?
Here. Yeah.
Yes. So, how do I know whether this means, what is the meaning of this exclamation mark?
This is the warning sign. You can find it over the FastQC website also. Okay, so there is a video tutorial also available
So
if you are
no,
interested.
so this is an unwanted sequence that may contaminate my data.
No, this is saying that there is a issue. There might be a problem with this sequence. Okay, so this output has some problem, but the red cross is saying that there is an issue, there is a problem, but it says that it might be a error, but it's not an error, it might be an error.
So we can overlook this exclamation mark.
Overlook, you can overlook, but you should not overlook. Like these errors, you should solve them,
So
resolve
it
them.
is better. So it is better we remove them,
Yes,
clean this.
yes.
So, sir, how do we know whether these overrepresented sequences could be important for us or not?
So
Because
could it be an
this is
overexpressed gene or something?
no, no, no. That won't be there. If it will find the adapter sequences or the sequence, the contamination sources, then it will show you. Okay. It will not
Okay.
show the overrepresented sequences of the genes. Like you suppose you have more duplicate sequences, complete duplicate sequences, and they have the possible contaminations or the adapter sequences, then it will show you the overrepresented sequences here.
Okay.
Okay.
So there is the reverse sequence also,
Okay,
reverse
sir.
file. Yes.
Sir, in this FastQC format, we look for only 3 parameters only, na, per base sequence and adapter content and that overrepresented sequences. Nothing else we look for the in this output. I think so. I get to know.
Yes, yes. Like most of the times we see that, but this is also important to find
Okay,
out
okay.
whether it is free of any
Matlab,
contamination or not.
matlab 3 parameters are the main
Right.
one.
Yes. Okay, so here you see the error rate is higher. The thread quality is much lower here in the reverse file. And the mean quality is also lower than the forward file, if you see here. So we will take care of this when we will run the Trimomatic to improve the quality of the data. Number of the sequences are also here having low quality compared to the forward sequences. This still has a problem in the reverse sequences. So we'll take care of this using the parameters in the Trimomatic. GC content is same, sequence length distribution has a problem here. That is an issue. That is an issue here because you see it says that most of the sequences are in the range of 251 base pair. So, but that is not the problem because we know they fall in the range from 235 to 35 to 250 nucleotides.
Sir, I have one question, but it is slightly outside this fast QC thing. When you before showing us the report, you showed us two lines where you are trying to retrieve read one and read two of the file. Correct, right?
Left command.
The forward and the reverse read.
Yes, yes.
Yes. So most of the time, so when we are searching online for means already published FASTQ files, we are getting the SRR files as it in the description is already paired. So is there a I have no idea, but is there a possible means do we do the FASTQC on that paired end file means single file?
Because there is very few help available for the Trimomatic over the command line, right? So you can open the manual, read it. I will paste the command here from that. Okay. So this is the command Trimomatic. You can install it inside the Anaconda or Conda, or you can also download the base and then. So if you download it from the GitHub or from the Trimomatic website, you need to run the Java minus jar and then give the path to the Trimomatic.jar. You will run this the same way, okay? Where your jar file or Trimomatic jar file is saved. You can install or you can change it by using the Conda environment. You can run here directly Trimomatic if you install it inside the Conda. So you can run here Trimomatic and then the paired end or single end. The data is single end or paired end. So we are going to use the paired end data. Then we can use the number of threads, that is 4. As 4 available here, I'm going to use the 4 threads. Forward reads, that will be the forward 100,000 fast queue. This will be the raw reads which we receive from the sequencing platform or sequencing service provider. Then the reverse read name, that will be the R or 2, then fast queue. So this is the reverse read. Then the output directory. So you need to create a output directory. You can name it anything. I named it here Trimomatic out. So you need to run the mkdir command to create this directory, and then you give the path for that particular directory. So run here Trimomatic out Mycoplasma pneumoniae forward paired sequences. This will be the output for the paired sequence. Then the second output will be here forward unpaired file. Okay. Then the third output will be the reverse pair, this one. Okay, first output will be forward pair, second output will be forward unpaired, then reverse paired, and then last output will be the reverse unpaired. And why it is like that? Because when we are going to use the forward and reverse files, there will be some proper pairs that will pass the filter. Then some reads will be good in the forward but not good in the reverse. So what will happen to those reads which are good in the forward? They will be kept in the forward unpaired. If the read is good in the reverse but not in the forward, they will be kept in the unpaired, and then the pair for that particular read will be removed from the paired file. Okay, so the pair file will have the proper pairs, forward, reverse, but unpaired files will have the reads which doesn't have the good quality reads present in the opposite direction of the reads. Then the filter values, or the values which you are going to use here, Illumina clip. So that will be the, if there is something available for these adapters which I showed you, I created, you can use, if these adapters are present there, using this option, Illumina clip all adapters .far file. So that will be the path of the file where you have saved your adapter sequences file. Or if it is present in the same directory where you are working, you can give the direct name here. Then look for the two mismatches when you align the adapters.far sequences. Then use the 30 quality score for the paired end sequences and 10 for the single end sequences. If they align good with the 30 or 10, single end and paired end and single end respectively, then trim them, because that is coming from this particular adapter sequence. This is the adapter removal in the Trimomatic. Next step is the leading. So remove the, from the leading, from the 5 prime, look for the quality score of first base. If it falls from 3, remove that. Then check the next. If falls from 3, remove that. Do it throughout the sequence. The same way from the trailing, remove the sequences from the 3 prime if they fall, the quality falls from the 3, below 3. Then the next step is the sliding window, right? Sliding window means it will take the 4 nucleotides in one scan and then calculate the average for that 4 nucleotide. If it falls below 20, it will trim the sequence there and then scan the next 4. If it passes the 20 quality value, it will scan the next 4. If it finds that it is not good, it will trim there and remove. Okay, yes.
So it is a paired end file. So why are you assigning the option Fred score 30 first 30 and then 10?
You
Because,
need to give it in the same format.
okay, both are paired end, so we
Yes.
have to. You said one is paired end and other is single end file.
If you run it like this, you will get the error. Okay. So we need to give the things here like this.
One paired
Yeah.
and single end. If it is not single ended also file, then also we have to apply.
You have to give this, yeah. Okay. So
This is for removing the adapter.
removing adapter and the low quality bases.
Okay.
Okay. So there is some problem. And then leading 3, sliding window 410, and then yes, crop 240. The total length of the sequence remove at the 240 to trim the overrepresented sequences after 240 nucleotides, and then head crop 19. Remove the first 19 base pairs because they were the overrepresented sequences. Okay, there is some problem. What is that? Trimomatic? No such file. Okay. So we have the 100 first two. Forward pair. let's create the directory that is Trimomatic out, because the error is directory not available to create the output data. We will run it again. Okay, it is done, and we can see out of 100,000, both surviving 92%, 92,973. Forward only surviving and reverse only surviving because their pairs were not good and removed from the data set. Okay, so the output is created inside the Trimomatic directory. You see, forward paired, reverse paired, forward unpaired, and reverse unpaired. Right? We will go inside the directory and then quickly run the fast QC minus out dir here only, and then give the input file name. I'm using the paired files only, not the single end or the unpaired ones. So now we are checking the quality of the processed data from the Trimomatic. First, what we did, we ran it with the for the raw quality data, raw data for the quality, and then we processed it with the Trimomatic using the parameters which we find out that should be used during the quality filtering. And then now we have ran again this, and we got the output here for the paired HTML and reverse paired HTML file. So I will open it and share the screen with you. Okay, so now here you can see now sequence length is 50 is the smallest, and the longest is 221 after the trimming. Okay, the quality has improved a lot for in terms of the average quality per base position here. The fret quality and base position, and you see this is almost 38 for the throughout the forward file. And then we had the problem for the per base sequence content. Now it is good. We trimmed it out, right? We don't have any error here. We still have the warning for the GC content, right? And the sequence length distribution, but it is not a big issue here, right? The same way for the reverse file, we have the output 50 to 2 to 1, 41% GC. The quality is much better than the raw data. And then we have the mean thread quality. You can see it is going up and then coming to the 39. Like very few sequences have the quality which is 39. Per base sequence content, it is good. Okay, one more thing, the adapter content here. It is also not present, which was present in the forward sequences. Okay, so this is how you can use the fast QC coupled with the Trimomatic. If you have the bad quality, you can process your data and then make it a good quality. Now this data can be used for the downstream analysis, like if you want to do some kind of SNP analysis or so variant calling, or you want to do some kind of genome assembly and then do the annotation process, then you can use this processed data, right? So I'm done with this quality control and introduction to the NGS dataset. And if you have questions, you can ask. I'll try to answer, and then we can wrap and close the session. Okay, you have some questions written here. Do you have questions?
Sir, what is base call?
Base call. I showed you in the presentation during the in layman term, you can say, suppose a nucleotide is fluorophore labeled there. Okay, when it is being incorporated, the with the you can say during the synthesis. Okay, so the detection is done by the laser, and then the fluorophore is emitting some light, and that is detected there and recorded. So that is the process of the base calling, right? That it is adenine, guanine, cytosine, or thymine. Which base is this? Based on the signal which was captured. So that is the process of base calling.
Dr. Surendra, are you going to tell us about the downstream processing of the sequence also?
Yeah, we will be going to the assembly process, and then in the next session after that, the annotation process.
Thank you so much.
So this demo in cleaning the data NGS reads were in Linux environment. Can this also be done in Jupyter or other Python environments?
Yes, you can do it, I think, but depending on the tools, if you can run them. Like for the inside the Jupyter, I'm not sure if you can run
I mean,
it.
there must be some packages designed to do that, right?
There must be, yes, yes. There are packages available. Yeah, you can do it there, or you can write your own script if you are good in the coding.
Excuse me, sir.
Yes.
Sir, for downloading of SRA file, it would be a single-ended or paired end. Because I was getting the paired end mostly from EBI database separately file. But the together file where from we get?
Yeah, you will get the single end file also if somebody has done the single end sequencing and they have deposited it. So it depends if the data is single end or paired end. It is nothing like you can download only paired end data. If the data is paired end, you will get the paired end. If it is single end, you will download the single end.
Sir, I was getting the paired end file separately, so I wanted to get it together from any database.
So from EBI, ENA,
Yes, sir.
I think, yeah, you were doing that. If you
Yes,
want
sir.
to use that, I think you can try using FastQ dump, and that will be good.
For separating the file.
Yeah, you can create the interleaved files there, sequences, or you can create the separate file.
So I wanted together file if possible.
Pardon, what?
So together file, that should be a together one.
Inside one file.
Yes, sir. Paired and both file. I should get by single click of paired and one file.
I don't know if you can do it there on the ENA, but you can try using FastQ dump.
I will try.
Okay, then.
Thank you, sir.
Thank you very much, and
thank you, sir.